What is RNAi? How it is discovered? And why it is important? These are the outlines of the talk. First of all, I will introduce about the RNA interference topic, and then we will explore the mechanism behind RNAi and gene silencing work, how it is discovered, and how in short span of time RNAi work got Nobel Prize.
There are several types of RNA. Some are coding RNA like messenger RNA and some are non-coding RNA. There are similarly different types of non-coding RNA, out of which siRNA and microRNA are very important that actually regulate the expression of the gene. So in this tutorial we will talk about what is the function of these microRNA and how it was discovered.
Here is the definitions of RNA interference. RNA interference acts on long double-stranded RNAs in a variety of eukaryotes to generate small interfering RNAs that target homologous messenger RNA, resulting in their destruction. This process is widely used to knock down the expression of genes of interest to explore phenotypes. Double strands of RNA, one of its strand is similar to the mRNA, the other is complementary, when introduced into cells, lead to breaking the mRNA, which is similar to one of its strand. The Jorgensen experiment on co-suppression in petunias, conducted by Richard Jorgensen and his colleagues in 1990, was pivotal in uncovering a phenomenon now known as RNA interference. In this experiment, researchers introduced additional copies of a gene responsible for petal pigmentation, the chalcone synthase gene, crucial for producing purple pigment, into petunias, expecting to intensify the flower's color. However, instead of enhancing the purple color, the flowers exhibited a strikingly variegated or even white appearance. The unexpected result was due to co-suppression, where both the introduced gene and the plant's endogenous chalcone synthase gene were simultaneously silenced. This finding was surprising, as introducing extra gene copies was thought to only enhance gene expression. The experiment revealed that plants could recognize and silence overexpressed or foreign genes at the RNA level, a discovery that laid the foundation for understanding gene regulation and RNAi mechanisms. Co-suppression in petunias is now understood as an early example of post-transcriptional gene silencing, a process widely used in plant research and biotechnology for studying gene function and engineering plants for disease resistance. The discovery of RNA interference, RNAi, in seeds elegans by Fire and Mello in 1998 was a groundbreaking advancement in molecular biology, for which they were awarded the Nobel Prize in Physiology or Medicine in 2006. Their research demonstrated the fundamental process of RNAi and how it could be used to selectively silence genes, revolutionizing genetic research and opening new possibilities in medicine, agriculture, and biotechnology. Here's an overview of their key findings in C. elegans. 1. Discovery of double-stranded RNA as a trigger. Injecting double-stranded RNA corresponding to a specific gene into C. elegans effectively silenced that gene. 2. Mechanism of gene silencing. They observed that after dsRNA was introduced, it led to the degradation of the messenger RNA, mRNA, of the target gene, preventing the gene from being translated into protein. This finding suggested that dsRNA activates a cellular pathway that targets and degrades complementary mRNA, thereby silencing the gene. 3. Systemic spread of RNAi in nematodes. Another fascinating discovery was that RNAi spread throughout the organism. Even when dsRNA was injected into only a small part of the worm, the silencing effect was observed across various tissues. This indicated a systemic effect of RNAi in C. elegans, allowing gene silencing to impact multiple cells from a single introduction site. 4. Specificity and potency of RNAi. Fire and Mello's experiments also demonstrated the high specificity of RNAi. The dsRNA only silenced the gene with a complementary sequence, leaving other genes unaffected. This specificity made RNAi a valuable tool for studying the function of individual genes by turning them off selectively. This research established RNAi as a natural mechanism cells use for regulating gene expression, protecting against viruses, and managing cellular responses to foreign genetic material. The implications extended beyond basic research, leading to applications in functional genomics, biotechnology, and even therapeutic approaches to treat genetic disorders and viral infections.